Okay, perfect. Thank you very much for the very nice introduction, for having me here as well. Let me just move this to the side. Okay, so as Theo already said, so my talk is going to be about parts of my project that I'm doing with Multiman. So just a few words about the Multiman Project. So it's a relatively big, really big project, and there's 15 of us PhD students that work on kind of different aspects of multilingualism. This is a picture of us, just to kind of put a few faces to the project. So what I wanted to talk about today was basically the fundamental question of how does the brain deal with more than one language at once. I want you to kind of keep that in mind for the rest of the presentation. Just as a brief overview, what we are going to do today. So I'm gonna give you a quick background into the study. Then I'm going to give some examples of how interference is manifested and how you've probably come across this already. Then I'm specifically going to talk about grammatical gender. And then also along the same lines, I'm going to introduce or recap, depending on what your background is. I'm going to recap electroencephalography or EEG before I actually move on to talk about the current study. Okay, so the background of the study is that if you have ever tried to learn a language as an adult or kind of later on in development, maybe you have experienced that you are having difficulties to kind of reach native-like levels. That was especially true for pronunciation, for example. And it is the case, it's a kind of well-established phenomenon that people who try to acquire an additional language, L2, L3, after puberty in adulthood experience these kind of difficulties. One of the explanations that was put forward as to why this might be the case was cross-linguistic interference. So this is essentially the interaction of the languages within the brain and subsequent kind of effects on the cognitive mechanisms. So when I talk about interference here, I don't necessarily mean the kind of negative impacts that on the negative connotation that the word has. So when I talk about interference, I mean both facilitatory effects and also effects that kind of go the opposite direction where they actually hinder processing. So as an example for cross-linguistic interference, so I think most of you are familiar with the cognate facilitation effect. What this basically means is that cognates, which are words that have a high degree of phonological and orthographic overlap across languages, are processed a lot faster compared to non-cognates. So if you think about it in terms of, so I have a German, Spanish example here. So if you have the word guitar in Spanish and in German, they are cognates. And they would be processed faster compared to a word like table in German and Spanish, which are not that similar. So basically what this represents is the kind of interference of the phonological representations. Because we can't really look at cross-linguistic interference in general, we try to focus on a very specific domain, and for us that was grammatical gender. So broadly speaking, grammatical gender is a way to classify nouns and to kind of form dependencies between articles and adjectives, for example. The nature of the gender system depends on the language itself and on the language family. I'm going to give the examples of German and Spanish because those are going to be the languages that we used in our study. So Spanish, for example, has a two-way gender system with masculine and feminine. And German, for example, for those of you who are not familiar with it, they have actually a third gender value, which is neuter. So now our study is basically based on some early studies that showed that native English speakers had difficulties acquiring grammatical gender in Spanish. And the difficulties were kind of not matched by their L1 Italian counterpart. So what that means is that the Italians had an easier time acquiring gender in Spanish, and the English native speakers were actually struggling a bit more. So if you think about the two language pairs that we have in these studies, so it was on one hand, it was Italian and Spanish, and then on the other hand, we had English and Spanish. Italian and Spanish are from the same language family, and they have a very similar gender system, whereas English actually doesn't have a gender system for nouns. So one of the proposals that was put forward by the authors was actually that for the Italian speakers, acquisition of the gender system was facilitated because there was kind of the influence or the interference from Italian into Spanish, and that's why they were better at acquiring the grammatical gender system in Spanish. Relevant to our study as well. So there have been a lot of studies on grammatical gender and cross-linguistic interference in general, and especially one of the populations that got a lot of attention was simultaneous multilinguals. So those are people that acquire language from birth, and typically they also have relatively high exposure and also high proficiency levels in the languages. So they were studied quite extensively. Then we have a second group, which are the sequential multilinguals. So those are typically speakers that acquire the language sometime after birth. The second language, sorry, after birth. They were also studied to some extent. So we have different kind of subpopulations of the sequential multilinguals. One of them are early sequential multilinguals. So they are characterized by kind of high exposure to the language from a young age, but larger, so from the age of 5 onwards. And they also have relatively high proficiency levels. One population that was studied a bit less were late sequential, highly proficient multilinguals. So those are people that acquire the additional language after the age of 12, so kind of a little bit further on in development, but they still have relatively high proficiency levels. The population that we were interested in, and it's a population that hasn't been studied very much at all, was late sequential, low proficient multilinguals. So they are characterized by kind of a low exposure to the foreign language. They have also acquired the foreign language in a later stage, and they still have kind of overall low proficiency levels because they're in the process of still learning it. So this is the focus. This is the focus of our current study, the population that we looked at. So our first research question was, okay, can we see or find evidence for cross-linguistic interference of the grammatical gender system in the population of low proficient language learners? I would like to open a little bracket at this point. So I would like to talk about how a multilingual brain could possibly represent gender. So there's two kind of opposing views. So one of them is the gender integrated representation hypothesis. And what it basically says is that if you have an item which shares the same gender across two languages, so in the example that I put here, it's again German and Spanish. If you have to retrieve or produce the grammatical gender of the noun candor, what happens is that the gender is shared between the two languages. So basically the same shared gender node is activated for the nouns that have the same gender across the two languages. On the other hand, if we are trying to retrieve the grammatical gender for a noun that has different gender values across two languages, for example, suitcase, so one is masculine in one language and some feminine in the other, different gender different gender values will be activated, but the gender values are still shared between the two languages. This is in contrast to the second hypothesis about the representation of grammatical gender. And it would be the gender autonomous representation hypothesis, which basically says that we have separate gender system and separate gender nodes for each language. So whenever we try to retrieve a specific word noun together with its grammatical gender, what happens is that we only activate the language specific gender node. The nice thing about the two hypotheses is that we can actually make predictions about what we would expect in terms of behavior and also in terms of the kind of neural responses, which I'm going to get to later. So the gender integrated representation hypothesis actually suggests that there is interaction of the grammatical gender systems between the first language and the non-native language. So what this also means is that we would see something along the lines of faster processing of gender congruent nouns compared to incongruent nouns. So what it means is that nouns that share the gender across two languages would be processed faster compared to nouns that don't. So here I have the example of forest, which has the masculine value in both German and Spanish, versus duck, which has opposite gender values in German and Spanish. On the other hand, the gender autonomous hypothesis wouldn't actually predict any interference between the gender systems, so we wouldn't really expect a difference between incongruent and congruent nouns. For now, research is pointing more towards what supports the gender integrated hypothesis. This is reflected in the so-called gender congruency effect. What it basically is the interaction of the gender systems, and it's exactly what we had predicted before. So it's basically that we process gender congruent nouns faster compared to incongruent nouns. There's another additional layer to this. So it might be the case that we can also see specific neural correlates or distinct neural correlates for congruent versus incongruent nouns in the form of P600 effects. And so before I kind of go on to talk about the P600 a bit more, I kind of just want to make sure that we're all on the same page. So I'm going to do a little recap on electroencephalography. Or EEG, much easier to pronounce. So basically what EEG is, it's a technique to explore brain mechanisms or kind of neural oscillations in relation to specific tasks or specific kind of cognitive processes. So what typically happens is that we have in kind of research settings, I have to add. So in research settings, we would have an electrode that's placed on the scalp, and then we can measure electrical kind of activity from the scalp directly with that electrode. What we're measuring specifically is postsynaptic dendritic currents. So it comes from those neurons, the nerve cells in the brain that are very, very close to the surface, which are the pyramidal cells. It's the kind of big cells that you can see in the bottom half of the graph. And this is basically, so the activity that we measure with the electrode comes mostly from those specific neurons. Yeah, this is me, just in case you were wondering. So this was in my very first EEG experience some time ago. Very long time ago, actually. So I just wanted to show you what we actually measure with EEG. So you can imagine us having a cap that is attached, or the other way around. So we attach electrodes to a cap. And what we then measure is essentially voltage amplitudes measured in microvolts. Here you can see that on the y-axis over time. So you can think of, don't worry too much about the units here. So you can think about this in terms of time. So what we basically measure is simply, okay, how much activity is there in the brain in a given time window? Well, over time, yes. And we don't only measure, traditionally we don't measure one electrode only, but we measure actually a few. So in kind of typical experimental settings, we would use about 32 to 64 electrodes. And then it looks something like this. So we actually have a lot of data from each individual channel. So what we measure is, in this particular study, what we measure is kind of the brain activity in relation to a very particular event. This is a technique called event-related potentials. And what it is basically, I give an example here. For example, at time 0, that is when you see the picture of a tomato, and what you have to do, and that's your task, you have to name the picture as fast as possible in your native language or your non-native language. So basically what we look at is, okay, at time 0, this is when the event happened, and then we try to see what the brain activity is like in the kind of in the time immediately after stimulus exposure. So that is a typical EEG setup. We have a set of ERP components that have in the past been linked to very specific cognitive processes. I give an example of a couple here. There's more, so that's not all of them. The labeling stands for the polarity. So if you probably noticed that this is a kind of a slightly older way of showing voltage. So we have positive amplitudes plotted downwards and negative upwards. So what the P1, for example, refers to is that there was a positive deflection in the EEG signal approximately 100 milliseconds after the stimulus has happened. The N2, for example, is the similar. So the N2 is a negative deflection around 200 milliseconds, and it was previously associated, for example, with inhibitory control. You can think of it in a task where people had to give a response versus where people actually had to inhibit their response. And for those trials, so that's when we would expect, for example, an N200 effect. Okay, so the ERP component that was of interest in this particular study was the P600. So the P600 is an ERP component that was previously linked to syntactic violations. And what it basically means is that, so I'll show you in terms of an example. If you think about the sentences, the cat will eat and the cat will eating. So the second sentence clearly contains a syntactic violation. And in the graph here, I kind of depicted the ERP responses or the signal that we would expect in relation to the first sentence versus the sentence that actually contains a syntactic violation. So you can see that the larger or kind of more positive amplitudes happen for the sentence that did contain a syntactic violation. So a P600 effect is basically showing a processing difference between constructions that contain a syntactic violation versus constructions that don't. And this is something that we looked at in our study. So there has been, to kind of close the circle again and come back to our study, there has been some preliminary evidence that suggested that there is a kind of different neuronal pattern for gender congruent items versus incongruent items in the trials that contain a syntactic violation. So what a study by Foucault et al. Found that there was P600 effect was slightly larger for gender congruent nouns compared to incongruent nouns. And this is something that we wanted to look at in our study. The P600 effect in late language learners is highly debated. So there's a lot of evidence that suggests that there is a P600 effect in highly proficient late learners. So what it means is basically that highly proficient late learners are able to kind of very easily discriminate between violations versus non-violations. However, the picture is not that clear in low proficient late language learners. So some studies found no P600 effect. Some studies found a smaller or slightly delayed P600 effect. So we wanted to know, okay, so what is it about this P600 effect in late language learners? Is there one, or can we not find evidence for a P600 effect? And that was our second research question. Okay, our third research question was is related to the to a joint effect actually of the cognate facilitation effect and the gender congruency effect. So if you remember before, I was saying that both have an influence on processing times. So cognates are processed faster than non-cognates, and congruent nouns are processed faster compared to incongruent nouns. However, in a lot of studies, this wasn't actually explicitly controlled for, so not both. So it was one feature or the other, but not both combined. So we tried to see whether there was evidence for a joint effect of the two features on the behavioral measures and on P600 amplitudes. And that was exactly our third research question. Okay, coming to our study. So we have, what we did was we employed a syntactic violation paradigm. I'll explain that in a second, to essentially explore the interaction of the grammatical gender systems between German and Spanish. Those were our two languages of interest. And what we were looking for was behavioral evidence on one hand for the interference of the gender systems. And then we were also trying to see whether we could find neural correlates of detecting those violations in speakers that were low proficient in the non-native language. And as I said before, so we were particularly looking at the P600 because that is the component associated to syntactic violations. So Our study included 33 native German speakers, and they had a B1, B2 level of Spanish. So we recruited them in Germany, so around at the University of Constance. And what we tried to do was to recruit them directly actually from Spanish language classes that were aimed at exactly the B1, B2 level. I'm not going to go into too much detail about the participants, but I'm happy to answer questions later. One thing just to point out is that the average age at which they acquired Spanish was around 16. So they were indeed late language learners of Spanish. Okay, so we tried to, so we gave them a number of different tasks in order to establish their linguistic background and get a little bit of more information about their proficiency, etc. We tried to give them the language background questionnaire, and we also asked them to do a kind of a vocabulary test in Spanish to estimate their vocabulary size. But what I'm going to talk about today is actually the main experimental task, which was the syntactic violation paradigm. This was the task that where we also measured EEG, and in particular, like I said, we looked at the P600. This is a table that kind of shows some example stimuli that we showed our participants, and it highlights the 3 different manipulations that we did. So first we manipulated violation type. So we had half of the trials that contained a violation and half of the trials that didn't contain violations. Then we manipulated gender congruency. So half of the trials again were congruent, the other half were incongruent. And the third manipulation was cognate status. Half of the trials were cognates, the other half wasn't. Okay, typical trial would be something like this. So people would see a single noun on the screen first. This was for the purpose of being able to minimize guessing. So we wanted to make sure that people are actually familiar with the noun before we ask them to do the kind of main task. So at this stage, people would say whether or not they have used the word before. Then they would see the same word in a very short kind of sentence or noun phrase construction. This particular case is a violation trial, so it contains it's the incorrect determiner. It's also a cognate trial, and it's also a congruent trial. And then we would ask people whether or not they thought the sentence was correct. And in this particular case, we hope that they pressed no. So this was the kind of main task of the experiment. We had specific hypotheses regarding our results. First of all, we expected faster response times at the behavioral level for cognate congruent trials because of this interference from L1, from German in this case. For EEG, we expected, first of all, simply larger P600 amplitudes for violation trials. So this would be evidence that people were in fact able to process there is a kind of difference in terms of neural patterns between violation and non-violation trials. And then we also expected the largest P600 effect to show for cognate congruent trials. And this would be evidence for interference and also the kind of interaction between gender congruency and cognate status at the neural level. Okay, just to talk you through my results. So we only looked at familiar trials. So we looked at trials where people said beforehand that they had used the word before. And then we also looked at only trials where people answered correctly as to whether or not it was a correct construction. What we found was 3 things. So first, we found that people were significantly faster for non-violation trials compared to violation trials, which was to be expected. We also found a significant difference between congruent and incongruent trials. So people were faster for congruent trials compared to incongruent trials. So this is kind of a demonstration of the interaction of the gender systems at the behavioral level, but I'll get back to this. The third finding was that we also had, contrary to what we predicted, we had people answering much faster to non-cognates compared to cognates. So we basically had this kind of reverse cognate facilitation effect, which probably you can't call it that anymore, but we had kind of, yeah, the opposite result to what we had predicted. For the EEG, so I'm just going to briefly explain the graph. So what you're looking at is the 4 different conditions. So it's congruency and cognate status here. And then what I've done is I've colored non-violation trials in blue and violation trials in red. And we have time on the x-axis. We have voltage amplitudes in microvolts on the y-axis. And what you can see very clearly, it's actually quite nice. So up until about 500 milliseconds approximately here for all conditions, we see that there is no real difference between non-violation and violation trials. And what this means is that we actually get people, so people don't really treat those trials differently. They don't make a difference at the conceptual level. But then what we see is that from 500 milliseconds onwards, which is the time window that we usually associate the P600 with, there is suddenly this divergence in signal, and suddenly the violation trials elicit higher amplitudes compared to non-violation trials. What I need to add is that we looked at, so we looked at familiar and correct trials again, and also we only had, we didn't look at the entire brain for this. We looked at, we chose a subsection of electrodes, which was kind of posterior, so kind of back of the brain electrodes. So the difference in the P600 window was significant. So what we found was indeed the amplitudes for violation trials were much higher compared to the non-violation trials for all 4 conditions in this case. So this is evidence for a P600 effect. What we also looked for was whether there was a difference, so whether one P600 was essentially bigger for one condition compared to the other, which we didn't find. So we didn't find that statistically speaking, the effects were different across conditions. What I need to add here, and I think it's really important that I mention this, so what we did was we did single trial linear mixed modeling for our EEG data. So typically in EEG research, you kind of have different options to analyze your EEG data. And traditionally, what people have been using is averages. So they kind of average across items, and then across items for each condition, and then across people in order to perform the stats. But what we have done is we have taken every single observation that we got from the recording, and we put it into a linear mixed model. So that way we kind of got around some of the limitations of the more average approach, and especially in light of the kind of unbalanced data sets that are a problem for average style analyses, we managed to get around this, and we think that we performed the analysis that was hopefully suitable for this data. It's a relatively new technique, so people have been pushing for it, but it's only finding its way into kind of multilingualism research a bit more. So just to summarize what I said, so from kind of pulling all the results together, we found interference of the gender systems also in our low-proficient late language learners to answer our first research question. So what we found was faster response times for congruent items compared to incongruent items. And this, we think, is reflective of the interaction of the gender systems. Secondly, what we found was a P600 effect. So it means that at the neural level, late language learners are indeed sensitive to syntactic violations. So if you think about late language learners, so it is the case that they look at syntactic violations, and then their brain goes, wait, hold on a minute, this is a violation. And that's why we see the difference in amplitudes. So that's also good news. That's really cool. For the third research question, we kind of we hypothesized about an interaction between gender congruency and cognate status to have an influence on behavior and on the kind of neural responses. What we found was at the behavioral level, gender congruency did influence reaction times. Like I said before, we found the opposite effect for cognate status, which is still something that we're thinking about. And what it could potentially mean. And then on the ERP side of things, we didn't actually find that gender congruency or cognate status were modulating the P600 at all. So it was basically statistically similar across the 4 conditions. So what it kind of means, to wrap up and put it back into the bigger context. First of all, what we present here is evidence for interference of the gender systems and an effect of gender congruency. So this supports the kind of gender integrated hypothesis where the gender values are shared between two languages. And what we also think is that what we're looking at is evidence that in early acquisition stages, like in the case of late language learners, gender congruency appears to be a much more salient feature compared to cognate status. And I think also a very nice kind of result of this study was that in contrast to some of the earlier studies, late language learners do show distinct neural patterns for syntactic violations. And all of these results pulled together, they're really relevant because, like I said, late language learners are somewhat understudied, and they are therefore relevant to kind of characterize the not only the cognitive but also the neural processes, especially during gender acquisition in this particular population. I think that was it. Thank you very much.